Hello, I am Alex Lipka. I'm at the University of Illinois in the United States, and I'm going to tell you about a simulation study that me and two of my colleagues are conducting. So, the rationale for the simulation study is that the basics of quantitative genetics is from Fisher 1918. As the number of variants controlling a trait increases, effect sizes get smaller, and subsequently, the genetic value underlying a trait approaches a normal distribution. So we might expect to see something like this. X-axis is the particular variant controlling a trait. The y-axis is the effect sizes, and we see that the effect sizes get progressively smaller and smaller and smaller. This has formed the basics of quantitative genetics for over a century, and there is a new theory that builds upon Fisher 1918 that this simulation study is going to look at. So here's a similar graph to the thing that we looked at on the previous page. X-axis is genomic position, y-axis is effect size. And if we say the ones with larger effect sizes are in red, and we have the smaller effect sizes in blue, then we can say we have an oligogenic component and a polygenic component. So, omnigenic model combines this with functional biology. It says that the red genes, the large effect ones, are the genes that directly regulate the tissue, that directly regulate the trait. And then the blue genes are the other genes that indirectly control the core gene through trans expression. So thus you have the core genes and the peripheral genes. Matheson 2021 built upon the basics of the omnigenic model to consider how genetic architecture behaves across subpopulations. So we still have core genes directly controlling the trait, and we got peripheral genes and perhaps some environmental factors that indirectly control the trait. And what we say in Matheson 2021 is that the peripheral genes form their own perfect storm network to indirectly control your core traits. Um, and they are different in subpopulations. So thus, if you were to do, say, like a GWAS for at a SNP that's nearby one of these peripheral genes, you might see different effect sizes in the two different subpopulations. So we wanted to test how likely the omnigenic genetic architecture is to evolve through the process of subpopulation generation. So we simulated a founder population where we had core QTL and peripheral QTL, red and blue. And then we did forward in time simulations. So then we had a whole bunch of different subpopulations where we simulate the same core and peripheral QTL. So we wanted to ask, do the observed core and peripheral quantitative genetic effects match what we would expect to see under the omnigenic model? We used AlphaSimR to do the forward in time simulations, and then we used simple phenotypes co-developed by me and my former postdoc to simulate our traits. Here's how we simulated our traits. Here's our DNA. Here's the plant phenotype. We use this statistical model to connect the DNA to our observed simulated traits. So we had core effects with normally distributed additive and epistatic effects. Similarly, we had peripheral effects that we simulated, and then we had the interaction effects between core and peripheral genes. If we expected the omnigenic model to be correct, we would have a figure that looks like this mock-up figure that I made. X-axis is generations of selection, and then y-axis is the genetic value variance log transformed for ease of visualization. So these six different lines depict kind of smoothing lines that we would expect the genetic variance components of our core genes to be. I expect them to be small and stable because core genes, the red genes we saw in the omnigenic model, would have similar effect sizes across different subpopulations. Meanwhile, I would expect the peripheral genes to have larger variance components because there's many more of them, and to be, and to vastly differ from one another. So, and that's because under the omnigenic model, we expect to have different transgene expression within subpopulations. And then I expect the interaction between core and peripheral genes would be something in the middle. So when we did our simulation study, here's one mock-up. This is not a mock-up graph. This is an actual result. We see that our results match what we expect on the omnigenic model, and we observed similar results in other scenarios. So I'd like to thank all these individuals and NSF for funding and Illinois Computes for the computational bandwidth to help us do these studies. Thank you. Bye-bye.